Now, coming on to the technique behind for the further development of Dolly, what was being done. If you study the diagram, see first of all, mammary glands from the clone mother. See, here is the clone mother. This is the clone mother. Mammary cells from the clone mother was taken, and the donor cell. These were called as the donor cell, which were having the normal growth cycle. The donor cells were starved. How they were starved? They were kept in a low nutrient culture medium. When the nutrient medium was very less in the culture medium, these cells starved. And finally, the nucleus was there. The cell was having the nucleus. Most of the cytoplasm has dried up because the cell was starving, and but the nucleus was intact. Now the donor cell was with the, actually it was starved, so there was no signs of growth seen here. Now it was being used. This is the first thing. Let us go to the other mother, that is the egg cell donor. Egg cell was taken from the other sheep, that was an adult Scottish black-faced Eves, and then where is my cursor? Okay, and then the egg cells are actually they are unfertilized cells only, so it was having cytoplasm as well as nucleus. With the help of a micropipette, the nucleus was removed, and now the cell is called as enucleated. When the nucleus was removed, it was an enucleated cell. It was being kept, and from the cell, the nucleus, the starved cell which we have received from the nucleus donor mother, so we have taken the nucleus from that mother and fused it with the cytoplasm, with the enucleated cell, by keeping it into a mixture of fusogen. Fusogen is the chemical which was used for fusing, and electric current was passed. Then the cell fusion was being done, cell fused, and they were giving rise after to fertilization. Why this is called as fertilization? Because actually two cells are mixing, two cells are combining. Then when the combination was being done, it was again electrically agitated so that it can start the cell division, it can show the cell division. So this zygote formed, we will be considering it as a zygote. So this zygote formed had the cell, has the cell content and the cytoplasm and the mitochondria of the egg cell donor, whereas the nucleus and the DNA of the nucleus donor mother. Clear? Now when the cell division was continuing, embryo was formed. Finally this embryo was implanted into another mother who was serving as surrogate mother. Then after the gestation period, Dolly was born. Okay, so this is the whole technique. You need to understand that how many mothers are there: one, two, and three. First mother was the nucleus donor whose mammary gland cells were taken, and these mammary gland cells were starved, and when they were starved, most of the nucleus content were lost and it was ready for the fusion. The other mother was the egg donor. Unfertilized eggs were taken, enucleated by a micropipette, and then fused with the starved cell. So nucleus was from one mother and cell content was from another mother, and the zygote was formed, agitated by electric currents, and it showed multiplication. The cells started dividing. When the embryo was formed, this embryo was implanted into a surrogate mother, and Dolly was born after the gestation period. That is it. This is the whole technique. You just see the picture. I'll be sharing the picture also later with you, and just go through it. Further, there are some important points I need to discuss with you regarding the cloning. See, you all have studied about the patenting thing, patents. So we are living in a world. I'll read it out for you because these are very important things and which will make up your thinking process, will make you think about all these things. We are living in a world where anything can be made exclusive just by claiming a patent or a trademark. Anyone can get a trademark or get something patented, but can a patentee claim a patent on a cloned animal? So, because Dolly was a cloned animal, so can that be patented? This question was posed by Keith Campbell and Ian Wilmut, the creators of Dolly the sheep, the mammal cloned from the adult somatic cell at the Roslin Institute of Edinburgh. This was the institute. Now, after receiving a patent on the method which was being employed by Ian Campbell and by Ian Wilmut and Keith Campbell in the creation of Dolly was SCNT, that is somatic cell nuclear transfer. So after these both two got this method patented. After receiving a patent on the method of cloning mammals, that is SCNT, somatic cell nuclear transfer, Campbell and Wilmot filed an application to claim a patent on Dolly also. Dolly or any other cloned animal which they have cloned or might be trying to clone. So this application was rejected by the patent attorneys and examiners. They explained that claiming patents for live clones was ineligible subject matter since one could not patent natural phenomenon. Okay, a panel of celebrated patent attorneys announced that though the cloning method can be patented, what they have done, SCNT was cloning method, so they can get the cloning method patented, and the product associated with the cloning method cannot be claimed for a patent. Why? So what happened in this case? Since Dolly the sheep or any other cloned animal were a result of DNA used from the identical species, that was a creation of nature. Genetically, the cloned animal remains identical to the donor parent, so it cannot get a patent. Finally, Dolly lived a successful life. Moreover, Dolly remained alive and well after her birth with a functional heart, functional liver, brain, and other organs, all derived genetically from the nuclear mother, from the mother whose nucleus was being taken, that is of an adult mammary gland cell, gave birth to the, and she also gave birth to four normal lambs. Dolly gave birth to four normal lambs. So she was living a quite successful life after that. The technique used to form her was later became to be known as somatic cell nuclear transfer that we have discussed. Somatic cell nuclear transfer. She was euthanized on 14th February 2003. Euthanasia, mercy killing. Euthanized by veterinary doctors. Why? Because around 6.5 age, generally the sheep can live for 12 13 years, but she was euthanized by the age of 6.5 because she suffered from arthritis in the hind leg joint and a virus induced lung tumor, which is a common disease among the sheep which are raised indoors. Dolly was very much pampered and always remained inside only, so that was a big loss, you can say, because uh, she cannot live for a longer duration of time. I'll be sharing with you some links for the articles. Study those articles if you are interested, and you will be benefited very much. So we are concluding here. Hope you enjoyed the class. Tomorrow, in the next class, we shall be continuing with another. By the time, read the articles and try to understand the topics. Thank you so much. All the best.